Immunology, differentiation of T cells. All right, so first and foremost, you have to remember that T cells are made in the bone marrow. Both T cells and B cells are made in the bone marrow, but where they mature is different. B cells will stay in the bone marrow to mature. T cells will leave the bone marrow as a precursor and then mature in the thymus. So it reaches the thymus as an immature cell. And as an immature cell, what do I mean by that? I mean that it's a CD4 positive and CD8 positive cell. So it has both of those positivities. So in the cortex of the thymus, it's both positive. They must undergo positive selection. And what does this mean? It means essentially that the T cells will bind self-MHCs, okay, to see if they have the ability to actually bind MHC classes, if they have the ability to work, and that's positive selection. Do you have the ability to work? Yes or no? That's positive selection. And that occurs in the cortex of the thymus. Then it goes to the second phase, and that's negative selection. Negative selection is to see if they have too high of an affinity for our own self-antigens, okay? So if they bind too tightly, then they should be killed off as well, okay? So positive selection is, can you bind in order to recognize MHC? Negative selection is, whoa, you might be able to bind too tightly and cause an autoimmune reaction later on down the road. We don't want that, okay? So keep that in the back of your mind. That's positive versus negative, and that's going to be cortex versus medulla, all right? Now, once you undergo negative selection, you get chosen CD8 positivity or CD4 positivity. You're either one or the other. CD8 is going to be obviously the cytotoxic. CD4 is going to be our helper cells. Now, the helper cells can differentiate into 4 specific T helper cells. It could be Th1, it could be Th2, it could be Th17, and it could be Treg, okay, regulatory T cells. All right, so let's start off with Th1 cells. Th1 cells are going to secrete IFN gamma and IL-2, which will help in activating macrophages and cytotoxic T cells, okay? So Th1, if you differentiate our helper cells into Th1 cells, that means we want to activate macrophages and CD8 cells, cytotoxic T cells. Okay, let's say it goes into Th2. Th2 is going to activate eosinophils and increase IgE. Okay, so some type of parasitic infection. Th17 is going to induce neutrophilic infiltration. Okay, and lastly is our T regulatory. T regulatory is going to prevent autoimmunity from occurring. Okay. And we're going to kind of dive into each one of these a little bit later down the road, but that's kind of the key things you need to take away. So these are just your CD4 T helper cells that can differentiate into certain subgroups or subtypes. So there's one kind of pathology associated with regulatory T cells, and that's going to be known as IPEX, immune dysregulation, polyendocrinopathy, enteropathy, and X-linked syndrome. All right. So what happens is that our T cells, or our regulatory T cells, activated regulatory T cells, normally produce anti-inflammatory cytokines like IL-10 and TGF-beta, which you already know prior helps attenuate the immune system. It helps relax it. Well, what happens in IPEX disease is that you have a genetic dysfunction or deficiency of FOXP3. If you have a decrease in FOXP3, then you have an overall decrease in T regulatory cells. And then that means you have an overall decrease in IL-10 and TGF-beta. So that causes essentially autoimmunity. So you'll see a lot of autoimmune conditions. It's highly associated in diabetes in male infants. So if you ever see a patient that's an infant, a male infant that has diabetes, that's so odd. You should never see a diabetic patient that's an infant, okay? If you see an infant male that has diabetes, then it's probably IPEX, okay? And that's a dysfunction of FOXP3, okay? Which leads to a decrease in overall regulatory T cells. All right, so that is kind of the differentiation of T cells. Now let's talk a little bit further about T and B cell activation. How do they get activated? How do they become mature? So first off, we have antigen-presenting cells that ingest and process antigens, right? So for example, our macrophage or any type of antigen-presenting cell, it's going to grab an antigen from its environment, it's going to process that antigen, and it's going to present it via its MHC2 or MHC1 class, right? Regardless of whether it's coming from the cytoplasm or from its environment. All right, so it's going to present it via MHC2 or 1. The T cell is going to be activated by binding to the MHC2 or MHC1 via the T cell receptor, okay? So that T cell receptor and the CD4 or CD8 receptor are going to bind to MHC2 or 1, and that's going to be the first signal to activate T cells. But we need another signal, a second signal, in order to fully mature the T cells, all right? And that is when our antigen-presenting cell has CD80 or 86, which is known on dendritic cells, which binds to CD28. Okay? And this is known as the interaction of B7 protein, which is just CD80 or 86. They don't really call it 80 or 86. They just call it B7 on the antigen-presenting cell, interacting with CD28. Okay? So you need both of those signals in order to fully activate the T cell. So the first signal is going to be from MHC2 or 1 binding to the T cell receptor and the CD4 or 8 receptor on the T cell. That's the first signal. The second signal is going to come from the antigen-presenting cell, specifically like dendrocytes, it doesn't matter, dendritic cells that are going to present B7 and bind to CD28 on the naive T cell in order to fully mature the T cell. So if you block that B7 CD28 interaction, you have peripheral tolerance, where essentially all the T cells they stay naive and you don't really have a good, robust immune response because you don't have any maturity of the T cells. Okay? So keep that in mind. Now, for B cell activation, first thing you're going to have is the T helper cell is going to be activated first, right? So you have to have the T cell helper cell activated first, and then the T helper cell is going to go and present the antigen to the B cell, okay? And so what happens is that we have like the CD4 T cell, the helper cell, with the T cell receptor, it's going to bind to MHC2 on the B cell, all right? MHC2 on the B cell. And then you also have CD40 receptor on the B cell that binds CD40 ligand on the T helper cell. What that's going to do is going to cause a release of cytokines. And those cytokines are going to act on the B cell, and it's going to cause class switching to take place, where essentially it changes its antibody formation in order to produce different types of antibodies to different antigens. Okay? So the B cell is usually going to produce IgM as its first antibody. Okay? So remember this process for B cell. First, you have the T cell binding it, okay, via T cell receptor and CD4 to the B cells, MHC2. And then you also have to have the T cell CD40 ligand bind to the B cells CD40. And that's going to cause a release of cytokines. And the release of cytokines is going to cause a release of B cell activated antibodies as well as class switching. Okay? So that's our T cell and B cell activation and differentiation.